Hello everybody. Today I am going to talk about various DNA sequencing techniques. So as you know, DNA consists of four important nucleotides: adenine, guanine, thymine and cytosine. In order to find out the order of the sequence, we carry out a process called as DNA sequencing. DNA sequencing methods can be categorized into different generations. So what we are going to talk about today is the first generation sequencing, which comprises of Maxam-Gilbert sequencing method, which is actually the chemical sequencing method, and Sanger sequencing method, which is also called as the chain termination method. So let us start with Maxam-Gilbert sequencing method. In Maxam-Gilbert sequencing method, we use chemicals in order to in order to get the sequence. So before I tell you how it is done, let us please see what are the sequences. So I'll just, so here we are using four different tubes. Each tube we will take the DNA to be sequenced. In the first tube, let us label it as A plus G. We will be adding formic acid. Formic acid will specifically modify only the adenine and guanine bases. In the second tube labeled as G, we will add dimethyl sulfate, which specifically modifies only the guanine residues. Then in the third tube labeled as cytosine and thymine or C plus T, we will add hydrazine. Hydrazine will specifically modify only the C and T nucleotides. Then in the fourth tube, which is labeled as C or cytosine, we will add NaCl along with hydrazine, which will modify only the cytosine, leaving out the thymine. And once all these chemicals are added, as the final step, in all the four tubes, we will add piperidine, which will break the sugar phosphate backbone near the bases which are modified. So in the first tube where A and G are modified, wherever in the nucleotide sequence A or G are present, the nucleotides would have got modified. So when you add piperidine, wherever the modification has happened, the sugar phosphate backbone will break. Before we do all this, please remember the entire Maxam-Gilbert sequencing technique relies on radio labeling of the nucleotide sequence. So the 5' end of all the DNA fragments which we are taking in all the four tubes should be radio labeled like P32 or S35. Hence, once this chemical treatment is done, next step is to run the samples which we have collected from all the four tubes in the electrophoresis tank, in the electrophoresis gel. And here you can see the table which shows us different nucleotides, what chemical agents can be added in order to modify them. So this is how the result will look like. Let us assume this was the double stranded DNA. We obtain first the single strand DNA, radio label it. For example, here they have added P32 to the 5' end. Once the radio labeling is done, we have to add this DNA into four tubes labeled as G, A plus G, T plus C and C. To all these four tubes now we will add the respective chemicals, and then followed by that we will add piperidine, which will go and break the sugar phosphate backbone at the modified nucleotide. And after that, we need to carry out autoradiography in order to get the bands. So once we run the sample in the gel, based on their molecular weight or size, they will run. The shortest fragment will run faster and the longest fragment will run slowly. As such, we will only see the bands, but in order to visualize, we will see all the bands. In order to visualize only the fragments which we have obtained after the piperidine treatment, in order to see only these fragments, you have to carry out autoradiography, which will enhance only the sequences which contain the radio label. So this is how at the end of autoradiography you will see the labeled bands. Now, how to read this, how to find out what is the sequence. So, please remember always in Maxam-Gilbert sequencing method, the sequence should be read from bottom to top, because the smallest means the first nucleotide. So, let us start reading the sequence here. So, you will see there is the same band present in both the lanes, lane labeled as C and C plus T, which indicates this band corresponds to C nucleotide. So, we will write the first nucleotide as C. Then the next band is present only here and not here, indicating that it corresponds to T. So, we have written T. Similarly, here you can see this band is present only in this and not present in the second lane, indicating it should correspond to A. So, next we have written A. Similarly, you keep reading. Once you obtain the sequence, now this is the sequence what we have in the beginning. Let us see if we have the same thing. So, CTA, CGTA. So, let us go back. You can see CTGA, CTGAA. Now, if you look at the band, this is what we have obtained here. ATGCATC. So, this is how Maxim-Gilbert sequencing is carried out. Now, if you look at the advantages, purified DNA, short fragments can be easily sequenced and it is a very straightforward method. The disadvantages is, first, because we are using a lot of chemicals, it is hazardous because of the extensive use of the chemicals, and also scaling up becomes difficult. That is, if the length of the DNA is very long, the process will be very tedious, and the getting a long read is very difficult. So, in order to overcome Maxim-Gilbert method, another sequencing technique was developed by Sanger. He got the Nobel Prize in the year 1980, Frederick Sanger, and along with his colleagues in 1977, they came up with a chain termination sequencing method, which is also called as Sanger sequencing method or dideoxy sequencing method. So, this is a PCR based method wherein we carry out in vitro synthesis of the DNA strand, and other than using the regular DNTPs, we use a special type of DNTP called as a terminator, which is actually a dideoxynucleotide. Now, what is special about this dideoxynucleotide? If you look at the structure of the dideoxynucleotide, in dideoxynucleotide, at the 3' position, instead of an hydroxyl group, it contains only the hydrogen. Hence, because of absence of this OH in the 3' position, when a new nucleotide comes in, there cannot be a sugar phosphate backbone formed because of absence of this hydroxyl group. The phosphodiester bond cannot be formed. Hence, there will be no more addition of new nucleotide once a dideoxynucleotide is added to a growing chain. That is why this method is called as chain termination method. So, here you will see the 3' OH group, which is necessary for formation of the phosphodiester bond, is missing. Hence, every time when in a growing chain, when instead of a normal DNTP, if a DDNTP is added, further chain growth cannot happen. The chain growth will terminate at that point. So, this process, that is why it is called as chain termination method. So, what are the requirements? Here we require a single stranded template. Template is nothing but the DNA that has to be sequenced. We require primer, obviously, because this is a PCR based method. DNA polymerase is required in order to synthesize the new strand. Based on the new strand synthesis, we will be able to identify what is the sequence. And we require the regular DNTPs, that is DATP, TTP, GTP and CTP. Along with that, we will also require 4 types of DDNTP, that is DDATP, DDTTP and so on. So, let us see how this process is done. As I told you, in the normal DNTPs, you will have 3' OH and 5' phosphate, and there is a phosphodiester bond formed between the 2 nucleotides. But in case of DDNTP, because the OH is absent in the 5' position, in the 3' position, there cannot be a new phosphodiester bond formed. Hence, chain termination will take place. So, to start with the process, here we have to take enough DNA which is required to run the PCR. We have to aliquate the DNA into 4 tubes, just like we did in Maxim-Gilbert method, and label them as A, T, G and C. In each of these tubes, we will add the regular PCR mix, that is, we will take the template DNA, that is the DNA that has to be sequenced. We will add the primers, forward and reverse primer. Then we will also take the regular DNTPs. Along with that, in the tube labeled A, we will add DD-ATP. Similarly, in the tube labeled T, we will add DD-TTP and so on. So, 4 tubes for 4 types of DD-NTPs, and then we will run the PCR. After the PCR is run, the product is then separated in the gel electrophoresis, and then from there, just like we read the result in Maxim Gilbert method from the gel, how we deduced the sequence, here also we can deduce the sequence. The only difference will be, whatever sequence was deduced in Maxim Gilbert method at the end was the sequence of the DNA that we took as the starting material for sequencing. Whereas, in case of Sanger method, whatever we are going to deduce, the sequence will be the sequence of the newly formed DNA. That means, whatever sequence is produced, that has to be complemented in order to find the sequence of the template, which is the sequence that we require. So, let us understand how that is done. So, this is what I was talking about, 4 tubes. Each tube contains the regular PCR mix. Along with that, in each tube we will take one type of DD-NTP. And then you allow the PCR to take place. Please understand, when the PCR is going on, your DNA polymerase is going to synthesize the new strand based on the template. Template DNA, if there is an A, DTP will be added. But when there is a T, the possibility is D-ATP can be added or DD-ATP can be added. It is a random process. So, in the first T, when there is a T in the template, instead of D-ATP, if DD-ATP is added, further chain growth will not happen. That means the chain will terminate. This is with respect to a single template, but in the tube multiple template DNAs are present. So, it is a random process. Hence, in one of the template, in the first position where T is present, if a D-ATP is added, further chain will grow. If DD-ATP is added, the chain will terminate. In another DNA template, in the first position, D-ATP is added, the chain is growing, but in the next position where T is present, if a DD-TTP is added, the chain termination will happen. In this way, different sizes of DNA product will be produced in each of the tube depending on where DD-NTP was incorporated during the process of elongation. So, based on that, when you run the sample in the gel, you will see different fragments. These are the fragments which are produced based on incorporation of the terminator and termination of the chain growth. Now, the reading process is the same from bottom to top, because bottom is the smallest, hence the first fragment. From bottom to top we will read. So, you can see this is A, T, C and G. So, the first smallest fragment is present in the lane which we have taken from the tube T. So, that means this is T. You can see here T. Similarly, second is from A. So, I am writing A. Third is from G. So, I am taking T. Like that you have to write the DNA sequence. This is the deduced DNA sequence of the product formed. Product is the DNA that has been amplified. Now, if you take the complement of that, complement of this, that will be the sequence of the template. So, this is how, using Sanger's method, using special nucleotides called dideoxynucleotide, you can identify or deduce the sequence of the DNA fragment that we have. This is known as chain termination method. So, as I told you, let us take a simple example to understand it more clearly. This is the sequence I have to be sequenced. So, first step is denaturing the sequence. I will get the sequence as a single strand, and then we use the forward primer. So, using the primer, I will take the template in 4 tubes. Each tube I am going to add a single type of DD-NTP. So, in this case I have added DD-TTP. So, this is the sequence. Now, T is added wherever A is present. You will see in the template there are 2 positions where A is present, in the first position and in the later position. So, in the first position, when the chain elongation starts, there are 2 possibilities, either D-TTP will be added or DD-TTP will be added. So, if DD-TTP is added, after this there will be no more further chain growth. So, the fragment size is only this much. With another template, here DTTP was added, hence the chain will keep growing, but in this position DDTTP was added, hence the chain will stop growing after that. Actually, there should be another third option where in both these positions DTTP was added, the regular nucleotide was added. In that case, the entire sequence will be amplified. So, this is the same scenario in this second case also. Here we have taken DD-ATP. That means wherever T is present, there are two possibilities. D-ATP can be added. If D-ATP is added, regular chain growth will happen. If DD-ATP is added, the chain will terminate. So, you can see in this position DD-ATP is added, chain growth has stopped. In this position, with another template, in this position D-ATP was added, hence chain growth happened, but in this position DD-ATP was added, hence the chain termination took place. Similarly, in the others. So, like this, different size fragments will be produced. Similarly, in the other two also, the same process has taken place. When these products are run in the gel, you will see bands of different sizes. These bands can then be easily identified. Now, in this case, you will see rather than seeing 4 lanes, we are seeing everything in the single lane. The reason being, I have used with each DD-NTP, I have used a specific fluorescent label. So, whenever the product is produced, it will have a different color. So, in this case, DD-GTP was labeled with the yellow fluorescence, DD-ATP is labeled with the green fluorescence, and similarly the other two. So, based on the fluorescent color, I will know what nucleotide it is, and I can write down here. Once I deduce the sequence, I have to complement it to find out what is the sequence of the template, and this is the sequence of the DNA that I want to sequence, find the sequence. So, now I have the sequence in my hand. This process is known as Sanger sequencing method or chain termination method or dideoxy sequencing method. So, you can see here 4 different DD-NTPs, each DD-NTP labeled with a different fluorescent label or tag. Then you carry out the PCR, depending on which position the DD-NTP is inserted, chain termination will happen. Once you have the PCR product, run it in the gel in the same lane. Based on the fluorescent which is detected using a photomultiplier, you will see different color bands. Those can be easily deduced to different nucleotides, and then you do a complement of that to find the sequence of the template. This is how Sanger sequencing is done. The advantage being it is much faster and more reliable and more accurate when compared to chemical sequencing method. The limitation is obviously if what we know with any PCR, if there is a non-specific binding of the primer, then the result of the sequencing may not be correct. So, through this we have learnt what is DNA sequencing, we have learnt what kind of first generation sequencing methods are available, and we have learnt the details of both the methods. We have also understood what is chain termination method and what is the role of DD-NTP in this method. This is the link of a video where you can see how Sanger sequencing method is carried out. So, with that I complete DNA sequencing. If you have any doubt, you can always get back to me. Thank you.